Hello, Donald Tormey, Executive Director of the Cancer Research Institute. And I think we've teed up this roundtable very well with the preceding 4 talks. And I think in this group, we all agree that 2010 and the beginning of 2011 are, I think, milestone years for cancer immunotherapy, and I think we're on an upward momentum. But I think we'd also all agree that these are incremental milestones that we've met, but we have a long way to go. And I think we'd agree that it is combination therapies among immunotherapies and with other modalities of treatments is probably where the field needs to move. And of course, combinations do pose a certain amount of challenges and hurdles. So the point today of bringing this roundtable together was really to bring individuals that represent the various stakeholders in the field of cancer immunotherapy. And this would be, academia, for-profit companies, the government, the FDA, not-for-profit institutions, and for-profit investors, to really bring this all together and to talk on the same page of identifying what we here collectively think the hurdles are that we're facing, and hopefully to come with some practical and actually applicable new models that we may need to put into place so that we can effectively all work together across these various stakeholder groups. And selfishly, from the Cancer Research Institute side, I do look to this, and with all of you involved too, in terms of really helping guide the Cancer Research Institute and the Cancer Immunotherapy Consortium in terms of how we can help to facilitate some of these new models or these changes and what we should be implementing and what we should be focusing on in this space. So that's the basic goal of this. And I think we'll first allow quickly everyone to go around the roundtable just to introduce themselves, what their affiliation is, a sentence or two about themselves so that everyone can understand we're kind of mixed up into these different stakeholder groups. So I'll start with you. You have to press this.
Steve Creekmore, I'm Chief of the Biological Resources Branch in the Developmental Therapeutics Program of NCI. We have grants and contracts that support biological development. We also have a manufacturing facility up in Frederick, Maryland, and put novel things into the clinic, some of which were on the various lists I saw. So we see things at the beginning, in the middle, and also a lot of things that didn't work out. And we're one of the players in, you might say, in translation.
Michele Maio, I'm the Director of Medical Oncology and Immunotherapy at the University Hospital of Siena in Italy. But I'm here mostly as Secretary of the NIBIT. NIBIT is the Italian Network for Tumor Immunobiotherapy that was established 4 years ago together with Giorgio Parmiani. Giorgio is the Director, the President of the NIBIT, which now involves about 40 groups that in Italy do cancer immunotherapy.
Good afternoon, I'm Steve Hody from the Dana-Farber Cancer Institute, the melanoma program there. And I'm also representing the Society for the Immunotherapy of Cancer, which is formerly known as ISPTC, and a representative from them in terms of this immunotherapy effort, and glad to be here. Thank you.
I just had the honor to deliver. I probably don't need to introduce myself.
Hi, I'm Mark Froelich. I'm a Chief Medical Officer at Dendreon, medical oncologist by training, and we're very interested in building on the success of ProVenge, both to expand our current platform as well as look at alternative platforms to move immunotherapy forward, as well as combine it with other therapies.
Nils Lundberg, Bristol Myers Squibb, and I work on new antibody drug discovery.
I'm Axel Hoos. I'm the medical lead for the ipilimumab program at Bristol Myers Squibb. I'm also a co-chair of the Cancer Immunotherapy Consortium, and my background is in surgical oncology, molecular biology, and cancer immunology, with some activities in biotech and pharma.
I'm Jens Vitomaschne. I'm the head of the immunological programs and clinical development oncology of Merck Serono, rather, let's say, mid-sized company, not really very big pharma. We have a couple of products in the pipeline, and the most advanced one is Stimulux, a vaccine developed in non-small cell lung cancer.
Hi, I'm Raj Puri. I'm the Division Director of Division of Cellular and Gene Therapies in the Office of Cell, Tissue, and Gene Therapy. There are 3 divisions within that office, between my division and Division of Clinical Evaluation and Pharmacology Toxicology. We evaluate all cancer vaccines, immunotherapy, gene therapy for cancer. And I want to send greetings from my office director, Dr. Celia Witten. I'd like to thank the organizers, Excel, for kind invitation, and I look forward to participating in this stimulating discussion. Thank you.
Jed Walchuk from Memorial Sloan Kettering. I guess that's my end user hat as a member of the immunotherapy clinical trial community, but I'm also here representing the Cancer Vaccine Collaborative of the Cancer Research Institute as a member of this organization that really was a pioneering network of sites that carries on immunologic monitoring and clinical trials in partnership with the Ludwig Institute. I can speak to the need for collaborative efforts among different sites to build on expertise, the importance of having access to reagents to really do these clinical trials. And I think we're at a very exciting time for a few reasons. One is obviously the recent approval of Sipuleucel-T and the exciting data with ipilimumab, but also the recently launched Clinical Immunotherapy Trial Network here in the US, which will also seek to unify some clinical trials to answer some questions in our field.
I'm Michael Becker. I'm the founder of MD Becker Partners, which is a strategic advisory and management consulting firm, works with life science companies to help them unlock value, create visibility, and access resources to grow their business. Prior to that, I was the CEO of a couple of biotech companies and also on Wall Street as a sell-side and a buy-side analyst.
Hello, I'm excuse me, Shalini Sharp. I'm the Chief Financial Officer of Agenus, formerly Antigenics. We have probably 2 forays into the cancer immunotherapy space. One is a cancer vaccine called Oncophage, now known as Prophage, which is an autologous HECHOC protein-based vaccine. And we also own QS21, which is the adjuvant used by GlaxoSmithKline in their MAGE-A3-based cancer vaccine programs. I've been at Agenus for 8 years, so I've been there for a lot of the history of what's gone on in the active immunotherapy space, and have seen and lived many of the challenges of trying to raise capital to fund all of these sorts of activities. So I believe that's the perspective that I'm here to provide, and thanks very much to Axel for including me in this panel. It should be very interesting.
And I'm Tito Fojo. I'm in medical oncology at the National Cancer Institute. Most of you don't know me because I don't work on immunotherapy, but I come to be here because Axel invited me. I'm actually the program director for our medical oncology program, and I tell our fellows that if they want to work on something that has a future, it ought to be immunotherapy. And so I believe in it. I became a believer because we did a 12-year retrospective analysis of all the prostate cancer trials at the NCI. And as James Gully knows, lo and behold, the PSA TRICOM trials stood out as something that were rather unique. So I'm interested in it, and I wish I were a greater part of it.
So having made introductions, we now have a pretty diverse group of people representing nonprofit organizations in this space, somewhat the investment community, academia, and industry. And we're hoping that we can tease out some weak spots that the space has, answer some pertinent questions, and hopefully provide some directions of what we do need to do next to continue to accelerate progress in this field. So having said in many presentations earlier that we're actually at a turning point for the field, there is now an emerging structure that could allow us to make progress faster and possibly produce reproducible success. So the question I'd like to pose to the panel, and that's the way we're going to structure this for the next 2 hours. Between Jill and me, we will basically moderate the discussion, and we also do encourage questions from the audience. So if there is anything that burns under your nails, please feel free to actually bring that up. There are microphones. And hopefully this will be then constructive. So I'd like to start out with the first question, which is directed to the whole panel, not to just one individual, about what your perception is of the state of this field. Now that it actually steps out of the shadow, more into the light, what is your perception that we need to do next? Where do you see it stands right now? What are the needs from the variety of perspectives that we represent at this table? So who would like to take the first crack at this pretty broad question? Okay. And I will be aggressive and take Michele for a first opinion.
I actually didn't volunteer, but anyway. Well, I guess the perception we all have also, I mean, from the meeting, from what we have heard in these 2 days, is that the field is definitely advancing. Now, we luckily have a number of agents that work, and this has not been the case for the past 20 years, practically, in the field. So, I think the perception is that we are all more optimistic about the field. The challenge, as Jill was saying, is for sure how to combine these agents that we know they work by themselves, and we didn't have actually to think about this question a few years ago, because we didn't have agents that would work in immunotherapy. So starting from the data that we have generated with these agents, mostly as single agents, I think the next major challenge and the perception of the community will be, now how do we combine them? And there the problems start, of course. I guess all the other questions will be around this very same topic.
Okay. A second opinion on the topic, Niels?
I'm not going to disagree that the entire field is rebounding. I think it's definitely on the upswing, and the various topics hit upon at this meeting are clearly evidence of that. I also won't disagree that combination therapy is the next big hurdle. I think it's a real challenge. I think that what is really needed is basic research. It's what I would like to sort of, as a representative from academia looking at, or from industry looking at academia, I'd like to see real basic research on the mechanism of action of immunotherapies. If we could understand the antigens that are required for a productive immune response to cancer in the same way that we understand the antigens that are required for a productive immune response to infectious disease, it would be a lot simpler for us to come up with dose and schedule. So there's big holes that academia needs to fill, and industry is not going to fill that.
I think that one of the challenges remains, and maybe a little bit of a view from an academic perspective, is that immuno-oncology, immunotherapy, not necessarily by those in this room, but the broader field, still is looked upon as a religion. And I mean, it really, I think there's some aspects where unless you have a mutation, you target that mutation, you see a 50% response rate, that's the only thing that people believe in. And that's kind of easy for someone to understand whether a drug is working. And the immuno-oncology aspect is a little bit more indirect. It's not a linear thought process. It's harder for some academicians or some investigators or some very smart people to get their hands around when they're not thinking that way, where they don't understand the field as much. And when trials are done where survival endpoints, which are important, are the only way to show whether something is truly working, which are expensive and time-consuming and laborious, that many people still kind of have this, I'm speaking for, have this perception of why you bother doing that kind of thing on our scientific review committee. So I think that's still, I would say that's still a hurdle. It's still a challenge, and absolutely the field is exploding. We're in the first chapter of hopefully a very long novel, but there's still many hurdles to overcome, and those perceptions, I still think are important hurdles as a community for us to be aware of. It affects scientific development in academic centers. It affects probably go, no-go decisions in financial decisions, things like that. And I think that's still a perception that needs more discussion.
So is this fair to say then, in other words, it's a credibility issue? So
It's a credibility. It's a why you're going through such a long process to get at the end when I have this TKI inhibitor that clearly gives me a response that's easy to do. I mean that I get that all the time. those kinds of sensations. Yeah.
So yes, Peter.
Yeah, I would like to fully support what Stephen has said. I mean, if I remember back 7 or 8 years ago, when we have had cetuximab in phase 3, and compare this now with Stimulvax being also in phase 3, the situation is completely different. At that time, when we have had cetuximab, there have been patients who really showed a clear response. There have been, I don't know how many requests and inquiries for compassionate use, even before we really brought this to the market. And now it's different with the Stimulvax, because the understanding of the endpoints, what we can provide in between, it's different. So I think we have to do a lot really to make the medical community to understand what is standing behind these immunological approaches. And I would even say that there is not a full acceptance yet available.
Yes, an opinion from the investment side.
Well, so I'm a Chicago Cubs fan, which means I'm an eternal optimist, and you probably got that from my presentation that I do believe that the active immunotherapy field is on the cusp of a bright future. I think one risk that we have, though, is managing expectations, and I think that was just kind of touched upon. But in particular, we use the term vaccine, and to a layman, to a patient, they equate vaccine with cure. And I think that's set very high expectations for the cancer immunotherapy field, and those expectations may not be met now in commercial reality. And as we start to be faced with charging $93,000 for a vaccine and $30,000 for signal transduction inhibitors, that suddenly people are going to expect or have in their mind a much different clinical outcome than maybe is supported by where the field is at. So I think we would do ourselves as an industry a favor by managing expectations about some of these advances.
Can we supplement this with an opinion from Shalini before we go to Tito?
Sure. Well, I would say that not only from the patient and clinical perspective, there remains some skepticism, but I'd say still in the investor community, we see sort of this dichotomy of responses to the recent news where half of the investors are looking for the next Dendreon and they want to know if we are it. And the other half of the investors, for some reason, remain unpersuaded by the recent developments in the field. And I think, as someone mentioned earlier, even from an investment perspective, it's much less risky to put your money behind a small molecule or monoclonal antibody, even today, with this field hopefully ready to explode. So I think translating some of that new enthusiasm into the practical outcome of funding additional work in the field remains a challenge.
So I'll just disagree a bit, since that's what you wanted in any case, right, Alex? Excellent. So one thing, I would disagree with the expectations things, because Avastin, even cetuximab, what they delivered on, nowhere met the expectations that were, touted. And at the end, everybody's fine with it. But with regards to why there's so much problem with, I thought about that. Provenge is no better, no worse than Avastin, and yet everybody worships at the altar of Avastin. And why is that? I thought about it, and one of the reasons is that by the time the Provenge data was presented, there was only a handful of investigators who had gotten their hands on this product. Whereas by the time the first Avastin study came out, there were thousands of investigators conducting clinical trials, and they've given it, they've seen it, they've experienced it. So I think that that's one of the major reasons. It's just that, you have to live it to believe it. And we're not doing that with some of these agents. And obviously, it's a different paradigm of sorts. And, you couldn't hand out Avastin to everybody. You could not hand out Provenge to everybody. But I think that that's one of the major problems. The other thing is, it takes you about 5 minutes into a immunotherapy lecture before you're lost if you're not an immunotherapy person. It took me 10 years to figure out what ELISpot was, After hearing Gully say, I finally said, for God's sakes, Gully, make a good slide, would you, So, that, whereas everybody, oh yeah, angiogenesis, yeah, I shut down the blood vessels, dead, So part of it is managing expectations, but I think at the end, expectations don't matter. If you have personal experience, then that's what carries the day.
So if I say that in a different sentence, education towards complexity of the novelty of immunotherapy for a oncologist that may not have that background.
Right. I mean, you talk about presenting antigens, and then all of a sudden you start to talk about restricted and this and that, immediately, right over everybody's head, so. And ipilimumab, how does that work, So most people don't understand it. But again, a TKI, yeah, I'm blocking some tyrosine kinase. There's a pathway shut down. I didn't need to understand a whole lot. Once you've gotten the concept, you understand all TKIs that come along the way.
So having said that, yeah, we'll come to you in one moment. I'd like to get one more point. When we define the field of immuno-oncology, beginning to do so, does that resonate with you?
Sure. I mean, it resonates with me. So, people talk about targeted therapies. And in the end, they're really not a whole lot different than traditional cytotoxic therapies. So, but it sort of identified something you could say, okay, I want to understand targeted therapy. So maybe people want to understand immuno-oncology. I think it's good. It's clearly saying that you are different than small molecules. Whereas targeted and cytotoxics, the overlap is so enormous as to be non-existent. But immunologic will never overlap with those. So I think that's clever.
Thank you. Jed?
I agree, and I think that not only is it a challenge in terms of the cognitive aspect, but there are also some feasibility issues with immunotherapy. In fact, I would argue that the two most dramatic examples to date of the ability of the immune system to treat cancer would be high-dose IL-2 and also adoptive T-cell transfer, both of which are either difficult in terms of the conditions where they have to be given to only select centers, or patient-specific, which makes them inaccessible by most members of the therapeutic community. I think that probably ipilimumab will hopefully be the first drug that will change that because of its ease of administration in sort of an outpatient setting. And maybe that will change it to something. Immunotherapy will be something that the more community-based oncologists will have access to and therefore learn more about. And I think as we have more antibodies, which oncologists are getting very comfortable giving, that will certainly make this less of a religion, which I completely agree with you, and more of, I think, the fourth conventional way of treating cancer.
Yeah, that's great. Ipilimumab may turn the tide, because that's going to be able to get into everybody's hands, and they'll be able to do it. And as long as they think of that as immune therapy, it won't quite be a vaccine, but if they're thinking immune therapy, then that will make believers of a lot of people. And by the way, yeah, you're right about the IL-2, not to monopolize the conversation, but it is true. It's a great modality. So are TIL cells and everything. I've been at the NCI for 28 years. I've seen Steve Rosenberg get 28 grand rounds every year. And I'm a believer. I've seen it. It works. But not in the real world. So it's a matter also of making something that is applicable to the real world. Provenge has some troubles with the real world. Right.
So we're living that right now. So we're in the second phase of the launch of Provenge, where we're finally increasing capacity to make it available much more broadly, and we're targeting obviously medical oncologists as well as urologists. And most of those people slept through their immunology lectures in medical school. So it's very sobering to me when I go to some of the genitourinary focused meetings, and you hear these beautiful lectures about the tumor microenvironment and the oncology, the chemotherapeutic treatment paradigm, and how we can build on that by adding additional agents to it, and with targeted therapies as well. And then there's 3 or 4 slides on when we have this great new product Provenge is approved, but it doesn't quite fit in with their lecture yet. And so yes, there's enthusiasm, but I think given the lack of familiarity with the mechanisms of action, there's just not as much enthusiasm. And the major competitors that we have coming down the pike are great advances for prostate cancer patients, but they're hormonal agents. They're hormonal agents that people have been using for decades. So there's a great degree of comfort and understanding with the mechanisms of action of hormonal agents. So urologists and medical oncologists are very comfortable with that. And when you talk about how would you use it even with a hormonal agent that's approved in a later setting, post-chemo setting, and most of them say, well, I would use it up front. I would use it as early as I could. But when you talk about an immunotherapy, I would stick right to the, exactly to where it's indicated. So it's kind of a measure of that comfort with the mechanism of action. I think that's an area that both industry and academia really have a shared and combined interest, is really educating people on the mechanism of action, because I think it's really going to be helpful for all involved.
I think I'd like to switch gears. I think we've realized in this discussion that we just took place that there's a problem in educating the general oncologist about the role of immunotherapies. But I think there's still other issues that have to be dealt with in terms of, I obviously deal with at the Cancer Research Institute with a lot with the academic world and researchers, and constantly hearing that in immunotherapy, most of what we're seeing tested are monotherapies, and monotherapies are certainly not the way they're going to work. They're suboptimal. They really need to be combined. There's lots of components within, not even combining with chemotherapy and radiation, but within developing an effective immunotherapy, you need a lot of agents to be brought together. And obviously these agents are held by different companies, somewhat always in silos. And I think the academic perspective, which of course I'd like to hear, I hear all the time, please give them, we need to get these, we need to combine them together, and how can this not happen? So I think, to me, I'd like to hear what the panel thinks about these hurdles. Do we need new incentives? Do we need new models? How do we get companies that own different proprietary agents to allow them to be tested, especially even in early phase clinical trials? Is there a different paradigm that has to be set up for that?
So in attempts to combine agents from different companies, so there's one thing is combining agents within a company or combining agents within different companies. And things that our community has discussed many times, and the two issues that always kind of seem to float to the top that maybe would be a launching point for discussion is intellectual property issues, and then another being a concern, and we have an FDA over here, there's always a concern, at least a perception, that if there's an adverse event that occurs on the trial, that it's going to be associated with both agents. And then there's a stigma against one agent, whether an adverse event is related to that or not. So I think those are some hurdles that have, whether they're real or perceived, are frequently discussed. And so on the flip side, I would say is that the dawn of this, of the first chapter, the dawn of this new era of therapeutics for patients, needs to be built upon, and we really have tremendous ability now that we understand not just the preclinical science, but the science in patients and people, that to take advantage of combinations and to maximize the benefits of all the different pathways and mechanisms involved. So it should definitely be done, but there are clear hurdles involved, and frequently the hurdles, at least in my perception, have dealt with lawyers and is one thing. So I think I'd like to get some feedback from people.
Yeah, what I think is that probably one way in which we could try to do combination studies in the early phases of development of drugs would be to not to try at least now to put two companies, two major pharmaceutical companies together, because that is difficult. I think that is, there might be also, there might be people willing to do it in each company, but then you go to the lawyers again, everything stops. Probably in this setting, the non-profit cooperative groups could help a lot, because as Jill was saying, small studies, small phase 2 studies, comparing different drugs, can be probably more rapidly done by cooperative groups, rather than trying to put giants together in order to treat 80 patients with a combination. And this is also, I guess it is the experience from several of us, that sometimes it's even difficult to combine drugs that belong to the same company together. So it's not a matter of putting together different companies sometimes, but it's even difficult to combine drugs that belong to the very same company. So probably the cooperative non-profit groups could help in designing rapidly developed small trials that could then drive larger studies made by the interaction between pharmaceutical companies. I don't know.
Thank you. Shalini, please.
Well, a couple of comments here. I think as a smaller company, there are a couple of reasons why we would love to do combinations. And in fact, we have a collaboration with Dr. Wolchok's lab looking at some combinations. But, you know, I think if the efficacy was phenomenal in combination, that's in our interest. So the incentives are aligned there. I think the other reason that we would have an incentive is that immunotherapies tend to work best in earlier stage disease. So the regulatory and development pathway for development in early stage disease is very difficult and long. So if a combination enables you to enter advanced disease, then there's a great incentive for companies to do that. But I think the downside and some of the disincentives are that if you are the incumbent, if you're already on the market, you have a commercial, basically product, you almost have only something to lose by being in combination, because if there is an adverse event or something to that effect, then it can only hurt you if you're already on the market. So the incumbent products, I think there's less of an incentive potentially to look for combinations. And then I think if you're not looking at incumbent products and you're looking at two investigational compounds to combine, then while there is some guidance on how to do that, it's certainly more daunting in terms of how to get to market.
If I may add on a bit and direct the question to Dr. Puri. As it comes to the safety concern that was articulated for combination trials of two investigational products, what is the regulatory perspective on this? That's part one of my question. Part two, the FDA has recently issued a draft guidance document for combination therapy of two investigational agents, which actually does not cover biologics, but it's a step in that direction. If you might be able to comment on that, where that might take us downstream, that would be helpful.
Yeah, I agree with Steve's, Dr. Hody's comment as well, that when you're combining two different agents and that adverse event, there are implications. And as a regulatory body and representing patients and public health, we are not going to ignore that adverse event. We're going to look at, and we're not going to indemnify one agent versus the other. However, we have our medical officers are very carefully will look at it, dissect out, and understand the mechanism of that adverse event, and try to identify one versus the other. So that's our answer for that. That's how we approach the combination therapy. Regarding the guidance on the combination therapies, we have been thinking as well as you all, and these kind of discussions are very helpful. We have discussed these issues at our workshop that we held in 2007 and 2009, and as a result of that workshop, we came out with a guidance document, which a number of you mentioned today, thank you, and many of you have provided comments that we are trying to incorporate. So we will continue to look at that and try to address these issues.
I'm not sure that gives much, your answer about adverse events. I'd like to hear from the industry. Does that, knowing that they're going to dissect it out, but you're still at risk. So is there any way around that?
I think the short answer is no. And maybe give a little perspective on kind of the pre versus post-approval setting. I think it's probably the greatest concern in the pre-approval setting when you've got a limited safety database and your product isn't well characterized. Once you have a larger safety database, you're approved, I think the adverse events are still a concern, clearly, but less of a concern, at least if the safety profile of the two agents, or at least your own agent, is well characterized. I think it's less likely that the FDA would take some draconian action for a single event, unless it was clearly mechanistically high probability of being related. The way that a lot of companies get around this, I think, with non-biologics is that they'll, if they have small molecules, they'll create a variant which is similar to but different to, so that you can use that in certain indications but not others, and that gives you some advantages in terms of how you price it, but also in terms of the adverse event profile. In terms of combining agents, I think in the pre versus post-approval setting, I think in the pre-approval setting, I've had these conversations, and I think the issues are exactly as Steve outlined. Safety, intellectual property, and then the third one I would say is control, just control of being involved and knowing what the data is before it gets presented somewhere. And in the post-approval setting, I think that the same conversations, the same issues get raised, but my response to those is, we're an approved product. If anybody wants to do this trial with our product, they can do it anyway. So it's a question of us being involved and scientifically involved or not. And so I think that it's much easier to justify it internally as well as for external investigators to do those things if you're an approved product. So that's unfortunately not the case for most of the products, but I think as we get more on there, it'll at least be a stepping stone to building combination therapies.
Yeah, Michael?
Sorry, and I don't have an answer for this, but I think there's a third component to add to this equation. We're talking about combining two investigational agents, but there's a lot to be done with using existing modalities in combination with immunotherapy. For example, there's an area study about radiation combined with a lot of the active immunotherapies, and that's based on some of the presentations I saw today. Again, as a layman, looked pretty tantalizing, and yet we're talking about leapfrogging that and going to the next level of combining two investigational agents when we're not even exploring existing modalities.
Well, I guess I would argue that I think some of the existing modalities, as even take ipilimumab, I mean, and I don't want to speak for Bristol Myers, but in terms of the percentage of patients that are responding to that, and I think scientifically or mechanistically, at least from what I hear from Jim Allison, combining this with something, and it could be radiation, but it also could be a vaccine of some sort. How do you get that immune response started before you take off the brakes? And so that I think there is still, to optimize active immunotherapies, I think there is still a lot to be done just in the immunological space, not necessarily combining with other things, but I'll defer.
If I may just add one more question here. There is one mechanism that I'm personally aware of that enables combination therapy investigation in a more safe harbor environment, not entirely safe, but improved, which is the CTAP mechanism. Is from the NCI side there any comment on that?
I'm not quite sure I follow the question.
Well, CTAP offers, of course, independent investigation of newer agents. They also do that for combination therapy. And for two investigational agents, and how it is in the room, the model that I have been introduced to previously actually encourages some of these approaches, but it's not entirely safe harbor. But it's a step towards getting away from some of the concerns that we just discussed. So is there a way to possibly even expand on the CTAP mechanism as it currently exists?
Yeah, I'm not sure what you mean by expand, but it's been extremely successful within the parameters that you've discussed. There's been a lot of willingness upon the part of many companies to, through CTAP and through the intellectual property agreements that have been established and data sharing agreements, to combine two agents, often two investigational agents. So the question is, and it's been entirely successful. The question is how to expand that outside CTAP. I'm sure we'd be happy to make our documents available, but you need a third-party broker. I think you need some independent third party. Now, the CITN, as it's coming up, would offer that opportunity as well. They'd be fully capable of entering into the same kinds of agreements.
Not to throw another monkey wrench in here, but one of the issues, though, I think the intellectual property rights that could certainly a CTAP type expansion, whatever, could help that. But one of the issues that I think we're facing right now in the field is how do you pay for these things? And to do an investigator-initiated trial to try to combine agents, the NIH money is a concern. No more R21 quick trials. We really go to pharma or other means to support these agents. And even if you're successful to be guided through a CTAP type mechanism to combine two investigational drugs, the question is how does one pay for this, and what's the role of academia or pharma or whatever power that be to get to move these things along? I don't have an answer, but that's a real life concern.
And even within pharma, study drug can be limiting, so please be patient with us.
guess that comes down to, is there a place for the not-for-profits in this? And can not-for-profits have I mean, again, should government be funding more of these early phase clinical trials? Should not-for-profit, like the Cancer Research Institute, I think what you found when Adam Colloquium presented our Cancer Vaccine Acceleration Fund, coming up with novel ways that we think we can raise new philanthropic dollars that we can, because when you get donor money as a person that raises a lot of money for cancer research, you have to show impact, and donors are getting more and more wanting to have immediate impact. So and they want to see impact on patients and results. So again, we think that the CVAF, and Jed maybe can talk to it as kind of an end user of this, is a way to help accelerate this.
Yeah, thank you, Jill. Oh, Chris.
Well, it probably didn't come through when I spoke what our belief is. Belief is, since the question of developing tumor vaccines is so highly complex, it obviously will take longer periods of times. That will threaten VC type of capital. It will take longer. We know that. In such a situation, not talking about your company, you are ahead, but the large-scale movement will take very long. And in such a situation, one must join forces. There's no other way. This means charity, this means governmental, this means universities, collaborative groups of all sorts. And this also might affect the regulatory hurdles. Does one wish to move ahead with selected centers having the skills and resources to do whatever G like or not? It's a very sensitive issue, but I do believe it requires long-term engagement and joining forces to really carefully develop. And if I say carefully develop, let me just mention what so far has not been carefully done. The regulators know that much better, and the companies as well. A drug should be developed according to pharmacokinetic and dynamic principles. This has not been done in vaccines. And there's a certain belief that one can improve on that level. In terms of the selective expression of the antigen of choice, that certainly might affect toxicity. And that is not so carefully studied in most cases. In terms of mode of action, you all know that. You have one antigen, and you love that, and you focus on it. But the mode of action in vivo situations is unclear. Whether that directly sets in motion an effector type of response, or whether that sets in motion bystander mechanisms to what we would believe expand pre-existing immune responses directed against mutational peptides. Completely unclear. Has not been systematically studied in vivo. Little in mouse, not in man. So I could go on with a list of essential things which should be done in academia and probably should be supported by all other stakeholders. But I do think there's no shortcut. And everybody is responsible to advance the thing.
So if I hear you correctly, and I think that's quite in line with the theme of our afternoon session, is that new models of collaboration are across a broader group of stakeholders, as we are all sitting here, is probably the direction we would need to take. Obviously, it's easy said, we need a new model. That model needs to be defined. But what we heard as the introductory talks to this roundtable already offers some ideas, right? What you presented, Christoph, was already innovative from an infrastructure perspective alone, not even talking about the science. CVAF is innovative. And building infrastructure just from methodology for the field, as CIC has done, also ties in with this. So the question is there any idea that this group can offer ad hoc or maybe offline afterwards, how we can continue to integrate for collaboration and build a model that is usable in the long run?
So that's an excellent point. And I think I wanted to give you sort of a concrete example of what the CVAF has done and the needs that it came from and also where it can go. So we had, along with other laboratories, been investigating a pathway that's a stimulatory pathway in T cells, and it's the GITR pathway. And we knew that many companies had clinical reagents. There was some trepidation of using an agonist antibody toward this kind of pathway because of this anti-CD28 experience and other agonist antibodies. And so we identified a small company, Tolerex, in Cambridge, that had an antibody but was utilizing most of their resources for another clinical program, actually in diabetes. And through the CVAF mechanism, we actually got that antibody out of the freezer there because of the resources that were available through philanthropy, in this case venture philanthropy, to do a really tripartite collaborative trial between Memorial Sloan Kettering and the Ludwig Institute and CRI and Tolerex together to really bring this one new reagent into a first-in-human trial. And so it really took the capital of the CRI and its skills in philanthropic fundraising in this particular area. It took the scientific experience that we and others had had in really validating this pathway as being important. And then it took the ability to generate a clinical grade reagent, which came in this case from pharma, from biopharma, to really come together to really treat the first human with this. And so this is a very complicated way to go, but I think that the pieces are in place. We just need novel ways to do it. And this venture philanthropy route is, I think, one viable way.
I'd just like to make one more comment on this point, which is that I hear a lot of discussion here about how to fund earlier stage clinical development, which I think is very important, because obviously that's where a lot of the innovation is coming. But as a company that is sort of middle stage or has even been late stage, I'd like to point out that there's also a gap here in terms of how to fund those activities, and that arguably because the execution of a pivotal trial is so expensive for a small to medium-sized company, that's potentially even a larger hurdle. And yet, I think when you look at government sources and venture philanthropy sources of capital, they're typically directed towards earlier stage activities. So that leaves the later stage activities really at the mercy of venture capitalists and public markets. And in that environment, you often see mainly the incentive is, how do you get to market? When are you getting to market? It's all focused on monetizing the asset as fast as possible. And that creates some of the incentives that were discussed earlier today, which force companies to take shortcuts. You have to sort of go as fast as you can without dotting every I and crossing every T in terms of developing all the earlier stage information you'd like to have before you go into a pivotal setting. So I think there is a gap here as well for how do you fund the later stage programs. And that is also very important because that's where you get the programs over the finish line and many patients can finally access them at that point.
You referenced the fact that, as was, I think, spoken about before, in terms of being pushed by investors to take the wrong model. I mean, going in pancreatic cancer, and I think this kind of brings us back to Axel's even introduction of the new, instead of doing phase 1, phase 2, phase 3, should we be talking about proof of principle and then moving on to a randomized phase 2? And what would it take, I'd like to hear, these proof of principle trials, what do those really look like in the immunotherapy space? What immunological data do you need for that? What is the signal to give enough confidence to move on to a randomized phase 2? And I'd love to hear that from all players here. Steve, you look like you have an answer. No? No.
I don't have any answers on this, but it's a great question. I have some thoughts. I'm not sure if they're answers. So I think one of the key principles that still need to be addressed in the field is, to make these go/no-go decisions, what are the right endpoints? And as you were just mentioning, in the expensive large phase 3 trials, you have survival as an endpoint. That's really hard to do for every agent unless you have another surrogate endpoint that will be acceptable from regulatory and safety and all the other issues. So in Immuno-Oncology, it may not be our traditional RECIST response rates. And so I think part of the field needs is more input, more investment, to use the term, for biomarkers that can help be predictable so that you could make these go/no-go decisions earlier, or maybe have things that are acceptable to regulatory agencies that would be a surrogate to a large phase 3 expensive survival endpoint. And I think that's certainly something that's needed.
But we should not expect a miracle. Maybe there will never be good biomarkers for immunotherapy. And you and I have had this argument before.
I'm a melanoma oncologist, so I typically am glass half empty, but we're now entering a different world, so I hope to be glass half full, and there may not be, but I think one could say it may not be a serologic marker, it may not be a pathologic marker, it may not be a radiographic marker, but there may be some combinations that could give you something that give you a hint to say that there's an X% specificity sensitivity that this patient is going to be receiving a benefit from this agent. And hopefully, I'm not saying it's easy, but that would certainly move the field much forward if there was something from a regulatory standpoint that was acceptable, that one didn't have to do a very expensive multi-100,000 patient randomized trial that you're waiting years to get answers for. It's a wish list at least.
Do you think that from your comment, that there are no immunological monitoring markers that you think we should be focusing on? Would you believe any of them?
No, I'm actually optimistic, but I don't want to raise false expectations.
But I think for this, it all depends what kind of drug you're talking about. And ipilimumab type of drug, you want a signal right away. You want some responses. For Provenge type of drug, you're not going to see that. So it all depends on, you're mixing, just like with the previous questions, is the combinations. Once you'd be able to convince anybody to combine anything with Provenge, it has very little intrinsic toxicity. If you combine it with something, you get bone marrow suppression or peripheral neuropathy and so forth, you're not going to say, Provenge did it. You're going to say it's the other drug that did it. Same with the PSA tricom. So I think you have to start discriminating if you're going to capture as your therapies not only vaccines but also ipilimumab type of drugs, then you've got to discriminate between those two classes of drugs. So, and ipilimumab, there better be activity in whatever you're doing, in terms of some tumor reduction or some tumor response. Do you disagree?
I think if we based all the immuno-oncology, whether it be on ipilimumab or vaccines, stuff like that, on pure response rates, that we wouldn't be where we are right now. So I think it's a little bit more complicated than that. And so I think that this potential that you're seeing, requiring the thing that's in seeing tumor shrinking all the time at high percentage is not necessarily going to be completely relevant to me. I mean, maybe, but not completely relevant.
No, I don't think that. I'm making the point that for vaccines that can't be, and that's why you were wanting to, what immunologic endpoint do we get? And then after that, it's religion. You believe in your drug, you got some immunologic endpoint, let's march forward. But for an ipilimumab type of drug, I think there should be some activity and not just the expectation that, well, I didn't see anything. Eventually, we see an overall survival.
So let me just redirect this slightly. So any immunotherapy, if antibody or vaccine or anything else, targets the immune system first before it targets the tumor. And there must be something that you can measure on the immune level first before you then get to your clinical outcomes. We have been struggling, accomplishing to measure these reliably and then connect them to clinical outcomes, which in part has to do with the learning process about the complexity of the immune system, certainly. But on the other hand, and that's what all these proficiency panels have been about, an attempt to deal with the technical issues of measuring. So first, knowing what you're measuring, if it's the right thing, and second, is there variability in the system that you have to somewhat correct in order to then have a reproducible outcome that can be connected to the clinical activity. And then once you are there, you can then start arguing, yeah, this drug should induce more responses than another, and should induce different types of clinical activity. I think what we all have recognized is that survival as an outcome parameter is something that immunotherapies are probably best measured with, but that takes time. You want something earlier for early development that is based on the true mechanism of action. So, having said a lot, some opinions on this point, where you see the value of the approach that we have taken, which is data-driven and pretty large community effort, to harmonize assays. That's one step in this direction. What else could we do collaboratively, and how do you perceive this?
I have just one little bit of bias. Like I said, I've heard Steve Rosenberg talk for 28 years. He's never used any surrogate. His endpoints are always tumor reduction. And he'll show you one scan after another where there's dramatic changes. So to think that immunologic therapies are only going to come later with activity and only show overall survival, whether it's XIO2, TILs, T cells that have been engineered to recognize any number of antigens, you can have as your endpoint for some therapies, not for PROVENGE, for some therapies, an immediate tumor reduction. So I've seen it over and over.
Nevertheless, though, we need, if you want to make some progress, you will not make progress if you think in short-term categories. So that's where the value lies for organizations like this. We don't have the business pressure of delivering a result in a bound time frame. What we can do is we can set a, let's say, 7-year time window and say, in 7 years from now, we want to have immune assays that are reproducible and usable to build biomarkers. That's what we did for the harmonization activities. In a commercial setting, it's not attractive. However, if we don't do it, we don't arrive. So somebody's got to help facilitate this. And my question again is, are there other things we can do? You can point us to that we can start, and then with some discipline, at some point arrive, instead of never starting, because it takes too long.
Perhaps I can say a word in favor of what we call it, guidance. Not there yet. Where are we right now? Most of us have used assets which are not harmonized. So we are on the way to harmonize technology. Then we usually restrict ourselves to look at reactivity against one antigen, which obviously is not enough to understand the highly complex interaction of those. It's a highly complex thing. It's a civil war. People like Thomas Wolfer have spent the life looking to this, and what you see that an individual patient recognizes multiple shared and individual mutational events. And if you go into that with an immune stimulator, and just look at the response to this particular vaccine antigen, you probably will miss something. And we need to do that. And in order to do that, we need an enormous amount of money and many participants in collaborative efforts. Before we don't do that, we don't know. And probably it's wrong. We have good reasons to believe that we are right, but we don't have the answer yet. And that's the firm belief that the T cell does it all. The antibody area, except a few of us, we haven't really systematically looked into. And again, there are multiple T cells that recognize the tumor-associated stuff. And if we cause a perturbation in the system, we don't know which ones are specific. So what I'm saying, we need to look at difference. haven't done that. We don't know it.
If I say this in other words, immunoguiding means identify the right parameters to measure in the right setting, and then harmonize in that context.
Must be a discovery phase, and then we eventually get to know something. But to say right now it doesn't correlate, it's pretty naive when we consider the complexity of the system and the complexity of the patient's germline.
I think Dr. Friedman had a comment.
I think one has to remember that these surrogates are just that. They basically are a way of making a choice to go forward, but you're still making a wager. You really don't reduce the uncertainty but a bit. So in other words, you might start out with a prior probability of about 0.01 that a vaccine would work, and with some positive indication, maybe move it up to 0.3 or something. I'll give you some examples. One is that we worked on. One is I think you had on your slide the BioVest vaccine. That was started out in our program here in Frederick with Biological Response Modifiers program with Larry Kwok. And at the very beginning, rather than an expensive correlative assay, what was put into it was a skin test. Basically, because each patient was getting a different oriented vaccine, and we were manufacturing each one, one could actually set up a skin test reagent with the same protein, and then see if and also a control protein. So you can see if you got a difference in simply the local reaction to that. And sure enough, there were a lot of local reactions generated to the id vaccine. And that was then in a sense, one got the courage up a little bit further to develop that further. By that, just because you're seeing a surrogate that looks very encouraging. However, by the end of the day, at the very end, the correlations with the skin test positivity or negativity versus whether or not the patient survived longer or shorter wasn't as strong as that. So in other words, it's a guide forward, a way of making a decision, but not necessarily a surefire thing. Another example is the surrogate measurements of we had two antibodies. And one was the cetuximab, and the other one was the chimeric 1418 against neuroblastoma. And the surrogate, that was the reason those were selected at the beginning is actually we were intending to do combination studies. Interleukin-2, because that at that time was a novel agent. We had acquired from a drug company that was getting out of an interleukin-2 development a supply of interleukin-2 that was ad lib. In other words, CTEP, or somebody could support those such trials. And both of them had excellent interleukin-2 mediated ADCC with NK. They were basically built in the same framework. So that was the reason, that was the initial part of the decision taking forward. The localization was interesting and so on, but at the end of the day, cetuximab was approved for, ironically, with a combination with other things it needed, which were part of the package coming in with the preclinical data. But the interleukin-2 part hasn't closed, that loop hasn't been closed. Since we're also developing interleukin-15 now, we want to actually look at something like that, or perhaps some other cytokine that might boost NK. But that's never, that part of closing the loop's never been done. With the neuroblastoma, there were, with the antibody, there were some responses, but not the kind of thing that you would be that interested in. And I think our colleagues that were in other parts of the NCI that were guided by a small molecule way of reasoning weren't as interested in it. So, however, as it turned out, in actually not that hard a pivotal trial, 160 patients on each side of the randomization for very good response post-treatment neuroblastoma, the high-risk group. One could easily, or one could see a distinct progression-free survival at 2 years. In other words, if you actually got going with a good combined trial early, that would have been along taking the pivotal status much earlier than 2010, which is or 9, which it was. So I think all these things basically are just ways of moving forward. They are not deterministic and they are not causal except by rough correlation. And you basically have to design a question to answer the next question, in other words, survival or something like that, as inexpensively as possible and move ahead. So I think really that's part of the issue. I don't think it's just a very expensive thing. It's a very clever thing one wants to do.
Case, do you have a question? Brian, can you?
I had a question for the panel concerning the wider area that was touched upon already by Christoph of immunopharmacology. All of the immunoassays we are conducting are indirect measurements of drug activity. But it now becomes possible with, for example, synthetic vaccines and monoclonal antibodies to actually more precisely trace where the drug goes, how it is metabolized, and what is needed for its activity, including, in the case of monoclonal antibodies, Fc receptor function, Fc receptor polymorphism, efficiency of complement fixation, affinity of the antibody. And in the case of antibodies, where what is the exact mode of action, maximum tolerated dose is no issue because we now know you can deliver it locally at much lower doses, exactly where it is needed, but which dendritic cells need to be targeted, where can we improve the targeting of the monoclonal antibodies to the right dendritic cells or the right T cells, etc. And so this is next to the new field of immuno-oncology. I would propose to have an area designated that I discussed actually with the pharmacologists in our hospital, a new field of immunopharmacology that will give this field more credibility, because then it becomes like other drugs. And I think we are capable with non-invasive imaging techniques with sensitive labels to monoclonal antibodies and vaccines to actually trace the fate in the human body of these novel compounds. And I think we have the ambition to actually start doing this in our own hospital, in a university setting, but I think it should be done preferentially in close collaboration with pharma. But I'd like to hear what the whether this resonates within the panel.
So, to build upon that, build a little bit what Axel had said. So, I mean, I think this community has done a tremendous job in trying to standardize LE spots and that, I mean, these key things to try to have some common ground. I think if we're going to have good potential biomarkers, we're going to try at least, we need to bring in some other disciplines too, including pathologists looking at a patient's first biopsy specimen for metastatic cancer, and is there something there that we could look at as biomarkers, morphology, staining, something like that. The nuclear medicine and radiographic advances, I mean, there's all these tremendous technologies coming out. Is there something that we could brainstorm and have some sort of way of thinking of non-invasive ways to predict outcomes by looking at whatever tracing molecules are working on in terms of immune stimulation or tumor response or something? But I think the field now needs to kind of go to another step where we're looking even in other disciplines besides our traditional ones of looking at serologic monitoring and such to take some other disciplines together to try to find a matter to find these additional biomarkers potentially.
There was a comment here from Jens Peter.
Yeah, I mean, coming back to the industry perspective, I think first of all it's very clear that we would love to have a biomarker we could rely on, because that would save a lot of time, and I think our management would be very, very happy if we could present this biomarker. But we have three problems. The first problem we have is usually we generate immunological responses, or what we call immunological response in peripheral blood and not in the tumor. That makes it very difficult because we know that these immunomonitoring results do not correlate with clinical results. Otherwise, we would need to go to tumor entities where we really have access to tumors frequently in order to check what's going on in the tumor. The second one is that the generation of hypotheses in terms of predictive markers or biomarkers is going in parallel to the development of the drug. So we start in phase 1. We may, if we are lucky, identify a first biomarker. We need to check in phase 2 if it is true, but in phase 3 then we would be able to really confirm this and not before, and this is the other hurdle. And eventually, I think it's not a regulatory question. If we go into phase 3 based on a biomarker, it is the risk for the company. Is the company willing to take a certain risk to go into phase 3 based on biomarker data? I can say from my perspective, the companies are not yet. They want to see clinical data just to be happy to go the next steps into the phase 3. That's still the case. And of course, everything what you mentioned is we have a group, the task of which is just biomarkers, nothing else. They are checking what are potential biomarkers. But again, we have lack of evidence or even confirmation that these biomarkers work.
So if I may add something to that, I fully agree with the perspective. There's a problem of circularity to some of this, which means if we say we look at responses in blood, but we don't look at response in tumor. Yes, that is correct. The problem with the responses in blood so far is that we actually don't know if these responses are meaningful because we don't know if we're measuring it accurately. Once we have to get over that hurdle, once we know we're measuring it accurately, we then have to look for surrogacy. We haven't done that yet. I said that earlier, I'm saying it again. It's a complex and long-term problem to get to the point of measuring something accurately. Of course, all of us want to publish results, we want to do a quick experiment and have something to show. But this is not what this is. It takes a long time. And I've said this at an FDA workshop. Same thing. People are pressured through all the environment that we're in to produce something fast. So, to come to your point, we don't know yet if the peripheral blood responses that we're looking at are actually meaningful or not. I wouldn't throw them out yet, because we don't know if we have measured them accurately. So that's number one. So we should fix that first, because we have the most experience there. We're the closest to actually figure this out there. And then the same issue will arise when you go to the tumor. You may actually have to then also make sure that what you're measuring in the tumor is reproducible before you then correlate with clinical outcomes. So both of them are needed. The problem, as you outlined, is exactly right, but it's complex. And again, this group of people, from the investment industry that needs to help us do this with some financial support, the nonprofits, academia, and industry, we need to work together to set up a paradigm under which this can be investigated, so ultimately everybody can benefit from it. So I'm just claiming for now, we're offering a model with the assay harmonization. It's just one way to do this, to provide an approach to reproducibility. And then you can test it. If it correlates, it's good. If it doesn't correlate, then you can throw it out.
Yeah, so I agree actually. Having a biomarker already, an immune biomarker that you identify that actually will be supportive, it will be added value to your clinical program. And these biomarkers don't just come up in the phase 3 clinical trial. It should be looked at every stages of this trial, including the preclinical developmental stages. So I think that there is a value of a immune response, immune biomarkers in this cancer vaccine clinical trial, because the mechanism of action is indirect immune activation. And the other comment the other chair had made about which is a music to my ears, was the randomized phase 2 clinical trial. I will also recommend it very highly, that you should look at your phase 2 data very carefully. I think someone presented that there are large number of failed phase 3 clinical trial in cancer vaccine area, and there are about, in my estimate, from my colleagues' estimate, about 8,000 patients have been treated in phase 3 clinical trials that have failed. So if you looked at it early and randomize it at phase 2 trial, looked at the data carefully, have a biomarkers or immune response and so on and so forth, perhaps you will succeed or not get to that stage where the large trial, which are very expensive, that failed.
Back to the assays, which I think is very important, both our area and the NIEID, which we actually have some vaccines. We worked with them and some of their, they have difficult, such as malaria vaccines and so forth. Nobody knows how to do that either. Find that the challenge where someone makes a challenge set and compares with knowns and unknowns, blinds and so forth, in to the outside labs that are being supported, just exactly how well do they stack up to some kind of a challenge set, has been very, I think, certainly has been very stimulatory to the people in the field that have participated in these challenges. So, in other words, the effort to actually try to challenge and to validate or qualify how well laboratories are doing in some key assays it would seem, if from another industry, if this were the electric power industry or something, that would be part of what they routinely would do. Or radiation therapy calibration, remember years ago when they found all the machines were out of calibration. What percentage of the amount of support that you would get for supporting these kind of assays do you think is a reasonable ballpark for paying for that kind of QC? In other words, are we spending too much, about right, or too little on those kinds of feedback and QC challenges?
You asking me?
Yes.
Okay, I say initially, because if you don't get a meaningful result without it, you should spend as much as you can on it so you can then work towards a meaningful result. If this is a hurdle towards measuring something useful, you have to do it. And then the investment is not a waste. It's an essential piece of the process.
To do what, or to continue?
Well, so first you establish your assay to any process that's necessary, if that's harmonization, validation, whatever it is. And then you decide, you use that validated process to investigate your biomarker.
Excellent. I remember not that long ago, I was sitting in the audience at a presentation when they were talking about the new, using the FISH assay for HER2 in breast cancer. And there was a lot of the similar types of disarray of data and incongruous laboratory results, but then a harmonization effort, much like what you've initiated here with the ELISpot, ultimately turned out to provide a reliable assay. And that was one marker in one type of cancer. I think the consortium has a real opportunity here to follow the same process you have done so with the ELISpot assay, and as a consortium identify what might be the key parameters that would need to be measured, and to repeatedly do that to help define what Chase has so conveniently put forward as immunopharmacology. Because if you look across the field of immunotherapy, we're starting to get enough of these things that they're falling into categories. And perhaps as time and data develops, we may be able to identify what these categories are and what key assay points are for the immunopharmacology within certain categories. And I think as the field grows, the consortium may have the opportunity to help define, with all of the intellectual wealth and experience here, what the key assays would be for immunopharmacological categories of different classes of agents, and then help define those to generate some common terminology and really build the field. And I think it's logarithmically more complicated than HER2 and breast cancer, but it's the same process.
I just want to say that I think this is one of the most important aspects of to advance the field of active immunotherapy, because today, despite FDA approval, despite randomized phase 3 studies showing improvement in overall survival, you still have investors, you still have patients, and you still have prominent physicians that don't believe the data because they don't see the shrinkage of the tumor, they don't see something that they equate to justifying the improvement in overall survival. And today, they still resist this new treatment modality. So without that, and without that question being addressed, I think there's a lot of risk.
I have one comment on this, and this is somewhat of a selfish comment from the CIC side, but we have observed 6-7 years ago when we started the process of building an infrastructure for the field that could hopefully make this more reproducible for success. First idea was we need to start at the beginning. So we look at biomarkers first before we look at clinical outcomes. Problem is there had been zero success. So at some point you need a success so you can fuel the rest. So we actually started from the back end and looked at improving clinical endpoints. I believe we have accomplished that by now. At least we've understood more. And now we have to go back and do that piece in between. From defining the product to looking at its biological effects, so then looking at its clinical effects. So we have defined product, we have clinical effects. That thing in the middle, which is the probably most complex, hasn't been adequately tackled yet. And we have some starting points, but we again need the effort of this community to make this happen. It's too big for one of us. We have to do this together. But your point is well taken.
I would just add that I think we need to manage our own expectations when it comes to biomarkers. That we now have 2 immunotherapies, one approved, the Sipuleucel-T, where absolutely right, it prolongs overall survival, but we don't yet have a biomarker. But that's a wonderful starting point. And with ipilimumab also, we have a therapy that in the large randomized trial improves survival. We don't know how to either select the patients beforehand or identify them during in any kind of reproducible way. But I think that we have to recognize this process as being very linear, that you first start with separating success from failure. And then you hone in on the characteristics that associate with success or failure. And I would argue that until now, we didn't have so many successes or failures, to make these comparisons with. And then I think we need to really separately address predictive biomarkers from pharmacodynamic biomarkers. Because predictive ones are going to be more difficult. It's going to entail looking at blood and tumor, maybe individual tumors within patients. But pharmacodynamic biomarkers, just as an example, looking at velocity of lymphocyte count change. Very simple, not very attractive from sort of the high-tech flow cytometry world that we live in, but may be very important. Start with simple things and go in a very linear way.
And for ipilimumab, wouldn't a predictive biomarker face a very high hurdle? I mean, you as a physician, you wouldn't want to deny a patient a drug that has a defined probability of a very durable response just because that patient falls into a pool where that probability is 30% lower.
Correct, absolutely right.
I think, Judd, I'd agree with what you say, and also the other comment that I would make is we've spent a lot of time, a lot of money, and a lot of patients, reagents, focusing on specific biomarkers that we think are the important biomarkers. And the end result of all that has been more or less no evidence for biomarkers that correlate with anything. So an alternative that is gaining increased momentum is to really not pretend that we know what really matters, but to systematically and comprehensively collect data about potential biomarkers. And at the early stage, we can think about, and all biomarkers are not the same. In the early stage trials, we want to think about biomarkers that perhaps give us evidence of bioactivity of the product. And that's very important because oftentimes we give product and we don't really know if it disappeared or if it was, if it had any function in vivo. And those can be as simple as a DTH effect, for example, just something that tells you the product had bioactivity. And beyond then, we can talk about biomarkers that give us insight into the mechanism or into the proof of concept. And all those build so that by the time we get to the later stage trials, we have some body of data that allows us to focus on a subset of biomarkers that might relate to efficacy in the end. So the critical notion is let's be comprehensive and broad about what we collect and not have preconceived ideas about what matters. Increased lymphocyte counts is just one example. And through that, we will be guided, if we look at it and we're a little lucky, into what will matter in the end.
Howard, please.
So we actually have Paul here, who, at least if I understand correctly, is offering a different way of looking at tumors. And after all, the tumor shrinkage that everybody's so concerned about, while it's dramatic to the eye, is still a very complex surrogate biomarker. I mean, there's tumor growth rate, there's tumor death rate, if nothing else, and there's variability from tumor to tumor site. So that's sort of been accepted, and the bar may be fairly high. Tito's discussed many times, as he referred to today, a fundamental change in tumor growth rate that's associated with the prospective vaccine that he mentioned. It seems to me that that is an adaptable system that could become a predictor of at least vaccine effect by looking at a change in the kinetics of tumor growth in some way as an early biomarker, and might even be able to predict, assuming the tumor doesn't change its stripes in the middle, the projected time to, say, progression-free survival, if not overall survival. But that's not adapted at all. So we may need more than just changing immune response criteria. We may need to fundamentally look at how the biology is changed by an immunologic intervention. I could foresee if that worked, being able to use that kind of tool to say, do we need one vaccine or do we continue to vaccine? If we stop vaccinating within 3 months, does the tumor growth rate change again? I mean, it's a potential parameter to look at that's fundamentally different and not based on a foundation that relies on tumor killing the way cytotoxic therapies do. We can't visualize the effects of the tumor. That's why we say it's not dramatic. I would guess if you had something like Tony Rebus's, dramatic pictures, watching T cells flux into a tumor, chewing it up, people might be impressed. Tito, can you comment?
I think we'll have to see if it gets validated. We're trying to do it prospectively in some studies, and if it works, then it's valid. And if it doesn't, it won't be. But certainly with an active chemotherapy trial, with a therapy that reduces tumor, we can predict when it's going to start to grow much ahead of the clinical observation. You can predict something is going to start growing even as it's shrinking, because you see the trajectory going downward, all of a sudden deviating, and you say, why is this happening? I'm still measuring a regression, and it's because something is starting to push it up that you don't quite measure, because you still see the bulk coming down, but you can estimate what the growth rate of what's eventually going to outgrow is. So we're trying with Lauren Wood and Berkowski to do a prospective study with their vaccine, and it'll be interesting. I mean, we're seeing some changes, but it's too early to say. But I think that a reason to have that, or it's one, the industry reason is to develop it. The other thing is, for something like Provenge, it sure would be nice if the physician had a quick readout of activity. It would give them a lot more confidence in the reagent that they're using. And I think it would be, good. I mean, we're in a society that likes instant gratification. That's why we like to treat and we like to see tumors shrinking. It would be good if we're not going to see that, and we have to wait long term, to know that there's some biomarker that would
Maybe I can just comment on our experience, which is that obviously a surrogate would be the gold, the holy grail. But I think just in terms of realistic expectations, that in our experience, we made a leap of faith early in Provenge development using peripheral immune response as a marker, and that guided us to the schedule that we used, and moving forward from phase 2 to phase 3. And in retrospect, then, we're actually quite encouraged that we haven't published this yet, but I presented it at the ISPTC meeting last year, beginning to see some really encouraging correlations between some of the things that we chose, for example, as our potency assay, degree of antigen-presenting cell activation by 54 upregulation, as well as the total number of cells in the bag, correlate very highly with overall survival. The numbers are smaller on peripheral immune response, but for each of the assays, so T-cell proliferation, ELISpot, and humoral response antibodies to the immunizing antigen, we see evidence of correlation with each of those assays in overall survival, kind of in the p-value of 0.05 to 0.1 range. which I'm relatively encouraged by given the relatively small sample size. So it doesn't correlate with each assay at each time point, but for each of them, at least there's one time point. So it encourages me at least that when we think about combination trials moving forward, that I feel relatively comfortable using those, that if we can augment the magnitude of that peripheral immune response, or if we're thinking about trying to improve the product in the bag, if we can increase the degree of antigen-presenting cell activation, that should be a good thing for the product and something that we would, the ones that do that the most effectively are the ones that we would think about moving forward with.
I think that I want to just share some of our experience. I think it's a great question. Also, the bigger challenge for us when we started involving the ipilimumab trials, at the Memorial, we collected more than 200 patients without a specimen. At the beginning, honestly, we don't know what should we monitor because we established all the ALSPA, tetramer, ICS assay. But the first question, which antigen? So because if you don't know which antigen, you should monitor. And so I think then we went back, look at it, just the absolute lymphocyte count. It turned out you see the change of lymphocyte count at week 7. It's a really fundamental, very simple assay, routinely clinical use. Now we further look at a subset, ICOS, and now nucleus antigen, but we don't think nucleus 1 as a one antigen, but the other antigen will be involved as well. So this opens a bigger question for us to gradually to investigate and probably have to try one by one then, based on all the knowledge we learned from all the current assays developed. Maybe some assay we never, pay attention to it. Maybe some later on come out, give a lot of, information. So for example, yesterday talk about the CRP, all this. We never thought about it before. So I think this is a bigger challenge for us. The society needs this kind of assay. I think we will have more and more topic and information shared. These platforms work together to understand what's exactly the immune system changed. So this kind of very effective the model. I think it's good, but I think we have to do it carefully, and that's our limited experience I have to share with you.
I would just say as an outsider that you also got to be careful that you don't set the bar too high for yourself, because nobody else has this. I mean, really, except for Herceptin, which is touted over and over again. And now there's a movement to, maybe we should have been giving Herceptin to women that have, 1+ and 2+ by immunohistochemistry and forget the FISH assay. So I think you got to be careful. And the other thing, he's telling you, I've got this, and it's a p-value of 0.05, 0.1, or whatever. What that's telling you is how complicated this all is. The reason is it's a probability, and it's not higher than that because it's one of 3, 4, 5, 6, 10 things that you could be measuring and that are important. And compared to angiogenesis, the immune system is 100 times more complex than is angiogenesis. And so I think that you're going to have to get lucky, and you might get lucky with one disease and one drug, and that probably or possibly would have no generalization beyond that. So you've got to be careful. And you also showed the drugs being developed. Why are they developing 9 in kidney cancer and 7 in melanoma and 9 in prostate cancer? All of a sudden everybody says, oh, these are immune diseases, and let's go after them. And I don't think a company's going to spend a whole lot of money and then say, well, that assay didn't work, let's stop here. So you got to be careful that you don't, be too strict with yourselves. So.
I would echo that. I mean, I think if you look at oncology in general, circulating tumor cells, for example, is an area that's a great degree of interest in. But that there is a plan, my understanding, to move that forward as a surrogate, but it's going to require multiple phase 3 trials with multiple different agents before the FDA would even consider that as a surrogate for regulatory endpoint. So I think we do have to have realistic expectations of what we can achieve with these.
So then, if I may, since we have about a half hour left for this panel, I'd like to get through 2 more questions if possible. The first one would be going towards funding. The concern of cost has been raised a few times now in different settings. Are there more, let's say, creative funding mechanisms that we could utilize to get to provide incentives for the investment community to help the immunotherapy field? So what would the investment community need to see, besides what we have already discussed? Is there something that the field can help foster that would actually increase funding? I'm turning towards our investment colleagues here, although there might be other answers from other directions.
I guess I'll start. This is a tough question because, I mean, to date, the traditional sources of funding, at least for our company, have been, the private and public markets in terms of, sort of just the well-known entities who invest in healthcare companies and biotech companies. I think one area of interest for me that has not been very well, I think, maximized yet would be the area of project financing. So you don't see a lot of mechanisms out there to fund a trial as opposed to funding a company, or funding a product as opposed to funding a company other than partnership with strategic partners. So I think that that's one area that is lacking, that has some potential. And there have been a couple of forays into this. There's a firm called Symphony Capital that was doing this. There are a couple of other firms that have tried to do this, and I think that there's potential there, but it's been a tough time for the biotech space in general. So those vehicles haven't had the opportunity to show the successes that they might have the potential to show. So I think that's one unexploited area. Another would be the venture philanthropy space. You hear more and more about this, and you see a lot more of it, surprisingly, outside of cancer. The Michael J. Fox Foundation, for example, other organizations, I think juvenile diabetes, there's some very large investments being made in venture philanthropy in industry, clinical trials, in other indications, and not surprisingly as much yet in cancer. And as I said before, I think when you do see it in cancer, it's less so in the later stage of clinical development. So I think that's another area where there's an opportunity. There should be a lot of motivation. A lot of people are personally affected by cancer. And there is a motivation from a philanthropic perspective, I think, to put some money behind this. And I hope it's just a matter of time before that emerges as well.
So thanks for going first, because it gave me a second to think about an answer to a very complex question, and I think I've got one. So you have a dozen phase 3 studies that have failed for reasons that probably relate more to how the clinical trial was conducted. Take, for example, the PanVac example where you were looking at an immunotherapy in a very late stage disease setting where you probably didn't have enough time to even see the effect from your treatment. If you could data mine the failed studies and fund that to a point where you've got a rationale for what to do to fix the failed study and move forward, I think that would be an area where you would have a lower investment hurdle, because now you're really just data mining. You're looking at patient data to try to tease out what went wrong. Or it might even be simpler than that. Maybe we just tested it in the wrong disease. But the issue that you'll have to address is that the IP for the asset has probably been damaged because of the passage of time. So you'll have to, it'll need to be a coordinated activity where you have buy-in from the regulatory body to extend the exclusivity period if you're able to find a positive rationale for moving forward. And if you can go to an investor and say, if I data mine this, or if I find a way to fix this failed study, I need a commitment that you'll fund the next stage of development. We'll fund the early data mining, but you've got to then provide the capital for the pivotal study or the randomized study. That may be a creative solution to some, I mean, there's so much data that exists out there that could be mined.
So a comment on that, actually. We had a similar idea in terms of data mining, what the field can offer. So everything in the public domain on randomized studies, randomized phase 3 trials with any immunotherapy, we actually put through the scrutiny of a committee. We had a workshop on it, and we published this. And some lessons that resulted actually helped us to understand the clinical kinetics better. That resulted in what we now call the concept of the delayed separation of survival curves. It takes some time for curves to part. If you look early, you won't see it. And we have had examples where that happened on almost all randomized trials with immunotherapies that seem to be effective. You obviously need a separation of curves in order to see that. GVAX had it. Almost any immunotherapy that has an effect, actually any that I'm aware of, has a delayed separation of curves. That concept, besides the statistics that you need to model it differently, came from a workshop that we conducted under the premise that you're suggesting. But we didn't do it to retrospectively fix what hadn't worked. We did it to extract lessons that we can apply prospectively. So for the next one, we designed this differently. So you're bringing a new idea here, which has some complexity, obviously, but it's worthwhile considering.
But it's a lot less costly to do that than to start a priori with a new immunotherapy concept and move that through phase 1, phase 2, phase 3. I mean, what would it take to resurrect GVAX at this point in time? You have a phase 3 study. If you look at the Halaby predicted survival for those patients, they were sicker than the other immunotherapy studies. What if you simply redid that phase 3? Wouldn't that be a more likely to succeed, less risk opportunity than I have a novel pancreatic cancer immunotherapy that I'd like to start to put into phase 1 trials? So it's a lower risk and higher reward opportunity if you can fix all the kinks. And it's not a minor issue. I don't discount that.
Yeah, if I just have to say one more thing. It's a very creative approach, and it would ask us to change many things in the current system, right?
It's complicated.
I leave it at that, yeah.
I want to follow up on that a little bit because it's a recurring theme with trials that we don't have enough money to really collect biospecimens and store them and such. Has there been a cost analysis done on the part of pharma to figure out how much on top of the bottom line would it really cost to do this in a systematic manner? Not even talking about the analysis on the backside, but just collect the samples. And what sort of thoughts are there in terms of the disposition of those samples should they be collected? And I can imagine pharma feels in a proprietary manner about those samples and there's issues to be resolved. I'd love to hear pharma's thoughts on that.
I won't be able to say anything interesting other than that we do keep track of the costs, and it's extremely expensive to collect and store samples.
Yeah, I think one of the challenges we faced is that it's when you're doing a large international kind of multi-center trial, as we're contemplating in an earlier stage disease, there's just a lot of logistics. And when you're doing it in Eastern Europe and whatnot, can you actually, serum is one thing, but if you're talking about cells and getting those cryopreserved properly, actually in the US trial, the IMPACT trial, we kind of changed from a optional sub-protocol to a mandatory protocol. And for ease, we were concerned that sending it to Dendreon and then sending to a CRO would confuse people. So we tried to do it to the CRO. And, even though it was a high quality CRO, almost all the cells we sent there were no longer viable. We're committed to doing that, at least in a portion of the patients in the phase 3 trials we move forward with. But I think that it probably needs to be a subset where we have confidence that the investigators know how to freeze the cells down properly, and we can be assured that the investment pays off.
Is there? I just want to know: is there a possibility here? And I don't know if pharma would be open to this. That if there was not-for-profit funding to support the getting of these biosamples and the storage and collection and making them, but then could you make them available to academic investigators to go into in-depth analysis of that? Or would still be your priority? If we were paying for it, would we be able to have access to distribute it among an academic consortium to look at?
I mean, I think it's an innovative proposal, and certainly if it were in patients that we wouldn't be doing otherwise ourselves, I think you could certainly make a strong argument that would be reasonable.
This is a great model. I mean, let's organize in a comprehensive manner the infrastructure so that data can be, or samples can be collected and stored for analyses, and give up a little bit of what you want to do and what you're afraid might come out of it for the benefit of having those samples. To my mind, that might be the most wonderful idea to come out of this session, not to diminish the others.
Just to follow up on that, so we're already taking one step in that direction. So, we have an investigator-sponsored trial program where we provide grant support for people who have ideas about how they want to use Provenge in combination with other agents. We recognize that a lot of those investigators coming forward with those ideas aren't necessarily immunologists or don't have the capability of doing those immune assays. And in part because we think it's important to have external validation for these immune markers, as well as for regulatory reasons for the investigator-sponsored trial program. The sponsor has to be kind of at arm's length from being involved in the idea of the trial, the conduct of the trial, and the analysis of the trial. And so we have people come to us saying, well, we would like you to do the immune monitoring, and we appreciate that we can't really do that. So what we're trying to set up, actually, Jed and I were just talking about this beforehand, maybe it's something we could do with the CIC, is to have an independent lab that would we provide reagents to, transfer some of our assays to, so that if somebody came with an investigator-sponsored trial proposal, we could give them a grant support to then pay this independent third-party laboratory to do some of these assays. Because I think as we look at these combination therapy trials with, in my view, immune response has got to be the primary endpoint for all of those kind of small research-intensive trials. You want to be able to look across those trials, and you want some degree of confidence that there's, reproducibility of the assays, and that, if you're seeing a better response in trial 2 compared to trial 1, that in fact is true.
This isn't a big pharma issue, though, for those 13 phase 3 studies that are failed. Those are small, broken biotech companies right now that would give their eye teeth for nominal funding to get to a point where they could take it to a pharmaceutical partner, to a venture capital investor, and monetize the asset. You have, there are a lot of companies that have learned a lot since the trial was conducted that they could move forward more aggressively, and it doesn't require tissue samples. It doesn't require complex analyses. I mean, GVAX is a great example, the BioVest example, now that they found the IgM component, NVAX. I mean, there are a lot of broken, and those are all small biotechs that, if you got nominal funding for, could move the needle. And then
I think nowadays we have learned a lot based on many things presented by AXA today. So what are the right endpoints, what are the right entities? We need to wait some time until we really can see an effect. I think I would assume that many of these companies wouldn't do these mistakes again. So I really think what we have to focus on is more the immunological focus on biomarkers, which can help us to predict or to show or to be a surrogate. The idea of collecting samples, whatever it is, peripheral blood or tumor samples, and having a centralized analysis focusing on specific questions which might help to identify biomarkers is good. I have to say we have to be realistic. I really wonder if companies would be prepared to give their samples of their clinical trials to a central storage or academia or whatever it is. I think if we really want to make that feasible, then we should rather focus on that we try to identify a group of companies who are willing to collaborate in this regard and who are willing to think about what could be the right strategy, and then come from this point that they investigate their samples in the context of this group in order to find out what's the right biomarker. I think this seems to be more realistic.
If I add to that, there is actually a mechanism that was established not long ago. It's in its mid-stage of development that came from the idea of the Critical Path Initiative, which is an FDA initiative, but it's called the Critical Path Institute, which actually is aiming to foster that kind of collaboration to build community-usable knowledge on biomarkers predominantly, but possibly other things. Raj, could you add something to this? Are you closer to that initiative?
Yeah, I think that's an initiative predominantly led by our colleagues in Center for Drug, but other centers, CDRH and CBER participates that. Yeah, so there are, many of you perhaps know that FDA has put out a number of guidance document called pharmacogenomics guidance document, and they are kind of touching this issue. And we had to address that biomarker issues. We have a mechanism called voluntary genomic data submission at the agency, and so where the data is not linked to any clinical trial. So we sponsor submits that data, and we review it. We do internal review, and we meet with the sponsors. And many times we had involving our colleagues across the pond at EMA to participate in those discussions as well. So provide the feedback to the companies on their biomarker development. And there are toxicogenomics biomarkers. People are looking into that, and particularly that hepatotoxicity and the renal toxicity biomarkers that in collaboration being identified, that FDA is working with the companies to further develop or promoting to development. Since we are on that biomarker, I just wanted to make another comment, is that that if you are considering to have a biomarker to eligibility criteria, for example, in your clinical trial, for example, looking at the particular protein expressing on the tumor target or a peptide or what have you, I will encourage you that you have interact with our colleagues at Center for Device Radiological Health to look at the analytical validation of the test before you embark at phase 3 clinical trials, and that because that will be required, we will invite our colleagues in the phase 3 discussions to look at the validation issues at that point. So I just say keep that in mind that other colleagues in different centers will be involved in that as well.
Raj, thank you for that comment. If I can expand on this just by one more step, FDA has obviously, in a very open and collaborative process, produced the cancer vaccine guidance. It's being completed now based on community feedback, as much as I understand it. Are there plans from your perspective, from OCTGT's or other agency divisions' perspective, to produce additional guidance documents that might help the community for immunotherapy?
We are, as Axel mentioned, we have received a number of excellent comments and from the community to cover that in the guidance document, and we are incorporating them as we speak, and we hope to finalize this guidance document and go through our legal system to get it, make a final guidance document. We are thinking about guidance documents in additional areas that I think, as the feedback we receive from here, from this group and others we interact with, to incorporate. And one of the areas I did mention to you about co-development of a drug and diagnostic, and that's hence my comment about analytical validation of your assay. We had, perhaps Axel, you're aware of that, we had put a concept paper several years ago, and we are working on that concept paper to putting out a guidance document at some point.
Thank you. And I think since we are running out of time, we have about 10 minutes. We'll try to actually complete this with some discipline in time, and go to the last question, which I would direct again, not to just one person, but to the panel at large, towards what future contributions do you believe, from the individual perspectives that you bring, do you believe that community organizations like CIC, like CIMT, like ISPTC, or now SITC, and others may bring to the table to address stakeholder needs? So from your direct perspective, what do you think we should do next? We've had a lot of fertilizing ideas in the last 2 hours, but if there's anything you would like to pinpoint us to, because we are in the process of planning our next initiatives, anything that comes from this panel can be considered.
Sort of factors into the remarks that were made both by Michael Becker and Raj Puri. It's okay. It expands the previous remarks, Michael Becker and Raj Puri. And your comments about harmonization. Basically, there are actually a community agreement on some of these issues. And we have approaches to solve them, FDA, and rightly so, and so on. And if you look at some other industries, there are examples where the government has a regulatory role and does that kind of thing and so forth. But the thing that is, and I think it gets to what Michael Becker was saying a bit, what I think the industry would really be benefited by, actually a formal standards organization, perhaps that could actually say, here is what these members are, and I'm talking about corporate members, and you could have non-voting government or something like that, but I think you want to keep that in, speaking from a government person, might be better to keep it actually at arm's length from a government agency, participation and so forth, but you really want to have, this is what the industry agrees it needs. And so if you sign in here, your company would be willing to do something, whatever it is that the board of directors would agree that would be, for example, pooling failed data, and you could feel free to find some way to share the profits that you might generate from that or whatever. You could be very imaginative, be totally free from being a government agency and just be an extension of the private enterprise groups. So, it's really ripe for that to happen. One of the things that's so striking about pharmaceuticals is so far away from other very profitable and highly intellectual organizations where they manage to pool some of their collective observations and actually make a profit out of what they find out. So I think that would be what if this organization could germinate that would be a good idea.
Just to kind of be a little bit philosophical, I think we need to continue to work together. I think we've put out a lot of what the concerns and what the issues are. I think we need to continue to be rigorous in terms of our standardization, our harmonization of how we communicate with each other about immuno-oncology and how we translate it into patients. I think we need to work together, the different societies, the community, to find ways to fund things better. We need to think about ways to approach our government in terms of funding. We need to continue open discussions with regulatory agency in terms of what the standards are, how we move things forward, not just quickly but safely. And I think the different efforts in terms of where basic scientists, clinicians, pharma, academia come together, we need to continue to communicate, because I don't think we all have the answers right now, but I think we know what the hurdles are, and to overcome the hurdles, we need to continue to communicate and work on them. And that's really my basic thoughts.
No, I think that putting together the efforts of the societies, networks, or whatever, will be crucial. As we have been discussing, in the US, you have two major groups that work on cancer immunotherapy. In Europe, the situation is different. It is much more fragmented, and the idea is to try to put these national networks within a general frame of network of European networks in order to improve the ability also of European groups to work together and hopefully to work together also with other groups outside Europe. I think that would be very important in terms of improving the field, besides what has been already said, interaction with the regulatory agencies and pharmaceuticals and so on.
Well, I could go along with that very well for another network in Europe. And I'm particularly excited about the possibilities to get samples from larger industry trials.
If I can just raise an issue that we haven't touched on today, but is one that I think will require close collaboration between industry, academia, and regulatory authorities, which is clinical trial design for personalized medicine. And as we think about targeting particular tumor antigens, the treatment paradigm that we have right now is we focus on particular tumors and do large trials that randomize patients. But I think as we think about personalized medicine, I think the ideal is that you screen patients for which antigens they express and be able to treat those patients with a personalized therapy. And we really don't have a clinical trial regulatory paradigm to do that with various different tumor types. And I wouldn't suggest that I have the answer here. I think it's a very complicated area, but I think it's one that we should focus on. I mean, I think if you think about what's moved oncology forward historically, it's been smart clinicians taking approved products and using them in different ways creatively. And I think with the current reimbursement environment, we're not going to be able to do that anymore. So we have to think about ways that we can work together to, in a very scientifically sound way, provide compelling evidence with a personalized approach.
I wouldn't advocate any kind of a change in direction. I think you're doing a great job. I think providing a neutral space, drug discovery is very hard, and it's getting harder. It's always been hard in immunotherapy. It's getting hard everywhere. I think providing a neutral space where regulatory, academic, and industry can get together and talk about these problems is absolutely critical.
Well, thank you. Then Jens Peter.
Well, I think we clearly identified that the development of active immunotherapeutics is different from the development of other drugs in oncology, and this is the reason why we have still a lack of acceptance of these drugs in oncology. This is also the reason why we might see, at least to a certain extent, a lack of funding. And the crucial point is really that we have no biomarkers identified, no surrogates, that the development is still very time-consuming and also costly. So therefore, I think the focus is, and the crucial point is really biomarkers, and I really think that we should work together here. The CIC, the CIMT, the ISBTC, all these organizations are the ones who are really very much reliable, and all the different stakeholders are willing to work together. I think what we could do is really to organize an initiative with big pharma and also biotechs in order to agree that we should, if we have a good idea, use our samples which we collected during the clinical trials to do a harmonized approach in checking immunological parameters. Perhaps that's a good way to do the next step then.
So I think that I agree with the general comment about participation of stakeholders of all walks of life is important, and as is also implicit in our guidance document development, we take your questions and try to develop guidance document to answer those questions. The other thing is I will recommend, I know perhaps most of you are familiar with the regulatory requirements and policies. And if you are not, whether you're an academic investigator or small company, make yourself familiarize yourself with the regulatory requirement. Failure to understand that can significantly delay in your development. It could be very expensive for you. So I can't emphasize it enough that for you to consider that. And the good news is that we are open for your interactions. We have pre-INDs, we have pre-INDs, we go out to the meetings. In fact, I'm here and 5 other colleagues are in the audience from FDA are able to interact with you. So I can just tell you that that we certainly request you to familiarize yourself with the regulatory requirements as we go along.
I don't have too much to add. I would just second or third the vote for more harmonization between the different organizations. I think we all have very similar goals, and I think if we can increase the collaborative efforts between the organizations, such as doing clinical trials together and therefore being able to share precious biospecimen reagents and answer questions with larger studies, we would make progress quicker.
So I think if you're going to look at some of these failed studies, that is going to require brokering between a lot of different entities, the government, from a regulatory perspective, IP to extend protection there. So that, I think your organization serving as kind of a broker or a consolidator, a liaison for that would be certainly an ideal role. And then secondly, everything that you're doing is applicable to the field of signal transduction. With all due respect, we don't know enough about that, and they're facing the exact same challenges that you are. So what some of the issues that you're tackling could be jointly or shared with others that, for example, the field of PI3K, which is suffering from exactly the same issues that active immunotherapy is. So if there's any sharing beyond active immunotherapy, I think that would be ideal as well.
So I just want to tease you on this point just for a moment. If we play this idea one step further, of let's say we as a nonprofit would take on the task of trying to broker the opportunity to look at data again, data mine lost opportunities almost, and then turn them back into opportunities. This is not an insignificant effort. It will require change of policy in some counts. It will require time and investment. where do you suggest we take the funding from to do that? We will actually have to change our structure in order to be doing this. We're not set up for this right now. It takes certain expertise, it takes time, it takes investment. So do you have a suggestion? Would the investment community be interested in supporting efforts like this? I mean, which are not at this moment directed to any specific cause. You have to set it up generically and then see what you can get your hands on.
Well, Shalini said it best when there are examples of this already in place. So the Symphony Capitals and project-specific financing could be certainly a conduit that hasn't explored this and would be well-structured or well-suited to do that. I would also say the industry itself would look at this. If you went to a big pharma and said, I'm going to come to you with an asset that's been vetted, I've data mined, I found out what went wrong. If you fund this proof of concept, will you agree to license the asset on the following predefined terms? Look at what Cephalon has done with their option agreements where they acquire a company. Seption is a great example. For $100 million, they licensed the opportunity to an asset. The company still had to do the clinical trial. The data still had to be positive. And what Cephalon was on the hook for was that $100 million, not insignificant. But if the trial worked, they had pre-negotiated the other $300 million to acquire the company. So if you went to Big Pharma with that type of an opportunity and said, we will have a governing committee or some way to vet these assets, but when we come to you, you have the option to say yes or no. But if you say yes, you will fund a pilot study to validate what we think is true, or maybe even the pivotal phase 3 study, whatever, or randomized study. But then you have to agree to option license the asset on pre-negotiated terms and work that out.
Thank you.
Well, I probably should have commented on this earlier because we did obviously have a phase 3 program which missed its primary endpoint, and we have managed to survive that, unlike some other companies. But we are currently working now in phase 2 on a new indication with the same platform technology. And so in some ways we're doing on a one company basis what Michael has suggested. You know, we're taking lessons that we have learned from our prior large pivotal programs and applying that now to a new indication. But I have to say, you know, while we are working on partnering opportunities, while we have been able to raise money and we hope to continue to do so, it is a challenge to find that funding, even when you have proof of concept data coming from a randomized phase 3 clinical trial. So, you know, I think it's a great idea, and I would hope that there will be some interest in funding that kind of data mining activity, but we are doing that right now, and we have found it challenging. You know, we're doing it, but it's very difficult. We hope to do more of it, but it has its challenges. But to go back to sort of the original format of just concluding remarks on what's next and what would be helpful, for me, I mean, I can't argue with any of I don't think anyone would argue with any of the comments regarding harmonization in terms of assay development and biomarker detection and things like that. I think a couple things I would add would be new sources of funding, so the ability to bridge between companies and access to government funding or philanthropic funding of later stage development activity, I think would be very helpful. You know, we're obviously already leveraging the traditional sources of funding in terms of financial markets and even non-traditional sources of funding from those traditional sources. But that would be helpful to tap new sources, I think. And then finally, you know, we have to be able to tell a story to investors that we can get our products over the finish line to market, you know, in the U.S. or other territories. So I think, you know, it's very nice to hear the remarks from FDA that they're willing to work with industry to smooth that pathway forward for new therapies. So I think if we can go to the market and say, here's FDA guidance, here's how we match up with that FDA guidance, and that guidance has evolved now to encompass these new technologies, you know, I think that would be very helpful for us specifically. I think the comments about personalized medicine, you know, to the degree that the regulatory paradigm can be developed further for the approval of personalized medicine, that would be extremely helpful. And I think the evolution of thought in terms of how to develop compounds in earlier stages of cancer would also be extremely helpful because generally speaking, immunotherapies tend to do better in that setting, but the challenge of running trials with overall survival endpoints in that setting is formidable. So some assistance from the regulatory side in terms of how to do that, I think would be extremely powerful for patients as well as the companies working in that space.
So just as an outsider, and I'll be brief, so I guess I was asleep in medical school when they were discussing the ELISpot assay, but I wasn't when they were saying that immune therapies would work best earlier in the tumors, when it was smaller and everything. And I think there should be a drive to do that. From our looking at Gully's data, it works best when you have a small tumor that is growing slowly. And that, I think, is a paradigm that will resonate with people, and I would hope that the field would move in that direction. Because the cytotoxic therapies that you showed in the 8, 7, 9, and 10%, if you look at those therapies in third line, second line, first line, it doesn't get any better. But immune therapies are likely to get better the closer you move up front and the smaller the tumor. So I would encourage that in a big way.
But it might not be true. It might in fact be that only once you have an inflamed tumor that it's going to be susceptible to immunotherapy.
Yeah, it might not be true, sure, but you ought to find out because the other therapies didn't deliver on that, but this has a possibility of possibly delivering on that.
Okay, so we'll take that as your closing comment for now. So having said that, I thank our panelists and the audience for being patient with us. I think this was a very stimulating discussion. It has given us some new insights. What we will do next with this information, we'll try to synthesize some key messages from this. Maybe we get to the point of writing a white paper, and you certainly will all see it. And you certainly will all get the chance to correct what might not be accurate, and possibly co-author this with us if you so desire. So in any case, we much appreciate the engaged discussion. Thank you.